Pharmapal.
Cross-docking of inhibitors into CDK2 structures Part 2. Hello everyone! Welcome back to our channel. Today, we're diving into the second part of structure-guided discovery of cyclin-dependent kinase inhibitors. This paper explores the fascinating world of drug design, specifically focusing on inhibitors of cyclin-dependent kinases, or CDKs. Before we get into the details, let's quickly clarify what CDKs are. Cyclin-dependent kinases are a group of enzymes that play crucial roles in regulating the cell cycle. They need to bind to a partner protein called a cyclin to become active. This activation is essential for cell division and growth, making CDKs important targets in cancer therapy. The authors aim to improve the discovery of CDK inhibitors by using a method called cross-docking. This involves docking a set of ligands, molecules that bind to proteins, into multiple protein conformations to enhance the accuracy of predicting how well these ligands will bind. Key objectives. The study sought to answer several important questions. Does using multiple protein structures improve docking accuracy? Can we predict binding affinities based on docking scores? What factors influence the success of docking and lead optimization? Now, let's break down the methodology used in this research. Data preparation. The researchers started with 150 high-resolution crystal structures of CDK2, the target enzyme. They meticulously prepared these structures by removing water molecules and ensuring the correct protonation states of ligands. Docking techniques. They employed two docking programs, Glide and Gold. Glide treats ligands flexibly, while Gold optimizes ligand torsion angles. Cross-docking, the ligands were docked into all 150 protein structures, allowing the researchers to analyze how different conformations affected docking outcomes. The results were quite revealing. Docking accuracy. The study found that using multiple structures improved docking accuracy, particularly with Glide. This was attributed to Glide's scoring function, which better accommodated the flexibility of the protein. Correlation analysis. The researchers assessed the correlation between the docking scores and the experimental binding affinities, IC50 values. They discovered that while docking accuracy improved, the predictivity of binding affinities was less reliable, particularly in scenarios with multiple protein conformations. Overfitting concerns. A significant point raised was the risk of overfitting, where the model performs well on training data but poorly on unseen data. This was especially evident in the cross-docking experiments. Next, we'll explore the key figures and tables illustrating their findings. Figure 1. Correlation analysis. First up, we have Figure 1. This figure presents the percentage of negative correlation coefficients across 150 structures from 5,000 scrambled datasets. As we can see, the distribution is fairly homogeneous, suggesting that the data is well randomized. This reinforces the idea that the correlations observed are statistically significant and not merely artifacts of random selection. Key point. A homogeneous Gaussian-like distribution indicates robust data integrity. Figure 2. Mean correlation values. Next, let's take a look at Figure 2. Here, the authors plot the maximum, minimum, and mean correlation values, sorted by mean correlation. This visualization highlights the variability in correlation across different datasets. The mean correlation intersects 0, suggesting that while some datasets performed well, others did not. Key insight. A small fraction of scrambled datasets outperformed the real data, emphasizing the importance of careful dataset selection. Figure 3. Comparison of experimental and scrambled data. Moving on to Figure 3, this plot compares the correlations for the experimental activity data to those of the scrambled datasets. The mean correlation for the scrambled datasets is close to 0, which is expected. However, the significant spread in the experimental data indicates that some protein conformations yield better docking results than others. Takeaway. 37% of grids showed statistically significant correlations, suggesting that not all docking results are equally reliable. Table 1. Monte Carlo optimization results. Now, let's examine Table 1, which summarizes the results from the Monte Carlo optimization experiments. This table illustrates how different grid combinations affect docking accuracy. The authors found that combining multiple protein conformations generally improves docking accuracy. Notable finding: the average improvement in docking accuracy when using multiple grids was about 5%, indicating that more data can lead to better predictions. Table 2, cross-docking experiments. Finally, we have Table 2, which details the correlation between the glide docking score and log IC50 at various RMSD cutoffs. This table shows how the docking accuracy varies with different cutoff values, providing insight into the optimal parameters for predicting binding affinities. Key observation: the correlation coefficients vary significantly with the cutoff, highlighting the importance of selecting appropriate thresholds for analysis. In conclusion, this study highlights the importance of considering protein flexibility in drug design. Researchers can enhance docking accuracy by utilizing multiple protein conformations, which is crucial for developing effective CDK inhibitors. However, caution must be taken to avoid overfitting when interpreting docking scores. Thank you for joining us today as we explored this fascinating research on CDK inhibitors. If you found this video helpful, don't forget to like, subscribe, and hit that notification bell for more insights into the world of drug discovery. Until next time, keep learning and stay curious.
Pharmapal.